So again, I would like to thank the scientific and organizing committees for inviting me and giving me the opportunity to present this talk. And also I would like to thank our colleagues in Sandoz Pharmaceutical for actually nominating me to present few slides on the biosimilars, and special thanks to Fatima Al-Hayati from Sandoz company. So let's start. As we know that since the introduction of biological medications over the past 3 decades, they have definitely made some revolutionary advances and discoveries in the field of medicine. We can definitely notice that we use biological medications in the management of several diseases, among which comes like the autoimmune or systemic autoimmune rheumatic diseases. However, despite all these advances that we have noted with the introduction and inventions of biological medications, there is a drawback that their cost does not allow all patients to have access to these medications. And if you look at Europe, for example, which is considered a continent with mostly first world countries, you can still note that quarter of these countries were not able to provide biological medications for their patients, mainly because of the economical status and the financial cost of this medicine. Now, a study showed that up to 50% of patients with moderate RA measured by DASTA 28 score, these patients in our rheumatology clinics, these are the patients that we usually start them on biological medications. However, here they were not able to start biological medications just because of the financial burden and the cost of this medicine. So having said that, there is something that I just learned a few days ago with respect to the biological medication. So for example, if you have adalimumab that you are giving to patient Ahmed, and you have another adalimumab that you are giving to patient Muhammad, and then there is a third adalimumab that you are giving to patient Fatma. So all these 3 adalimumab, also they are like adalimumab, however they're not completely identical. So the first patient, who is Ahmed, will be taking a batch of adalimumab, which is similar to the second batch in form of the sequencing of the amino acids. So the same amino acid and the same sequences. However, the sugar molecules that are attached to these amino acids differ. So the adalimumab that is given to patient Ahmed compared to patient Muhammad, compared to patient Fatma, they're not completely identical. So I would like to say that they're similar in their structure, but they're not 100% identical. And this actually, led to several, manufacturing processes for each of the biological treatments by the companies producing these biological medications, after their approval, which unfortunately led to an impact on the analytical variability. However, you can see in the graph here that how many times rituximab required manufacturing changes, for example 20. Look at, for example, infliximab, and we know that infliximab is one of the early biological medications that we have been using in the practice. However, it required several changes to help attenuate some of the undesirable side effects of these medications, such as injection site reaction, infections, and so on. Which means that these biological down the journey of their usage throughout the past few decades required several changes that do not make a biological identical to another biological. And here comes the definitions of biosimilars from both the EMA as well as the FDA, where they can simply say that biosimilar medications, in a way or another, they are similar to the originator because they still have the same kind of consecutive batches that the originator medications have among themselves. And as of like the EULAR ACR recommendations, which were proposed like few years ago, they stated clearly that if there is a medication that can be used with like a more cost-effective kind of way, then it should be used instead of like a biological treatment. And there is one thing else that I learned few days ago about like the medication, that the way that we assess like the structure and functional relationship is with the help of some of the following factors, such as the primary structure, impurities, biological activities, higher order structure, as well as like the post transformational modification, and then combinations of the attributes. So spending some time on this slide might take us back to the medical school, where we used to study like the pharmacokinetics and pharmacodynamics of these medications. However, at the bottom line of this that I would like to mention, that each medication is not completely identical to the other medication, especially when it comes to the biological medications.
Recording in progress.
A biosimilar also apply to this role. Now, there is one more difference between the originator compared to the biosimilar in terms of like the development and the scientific evidence that we can originate from these medications. In the originator development, it usually starts like with the analytical, then non-clinical, then like the pharmacodynamics, pharmacokinetics, so like the different phases of the clinical studies, and then we go on to the clinical part of the study, which is like the phase 3 study. So the major goal behind like development of the originator is to measure is to determine like the clinical effect of that medicine. When the biosimilar is produced, it is produced in the same similar fashion, molecules, pharmacokinetics, pharmacodynamics of the originator. So there is no point of starting the clinical trials of the biosimilar in assessing the analytical part. The major goal is to assess the biosimilarity, so we usually start like immediately with the clinical part of the study. Then that is followed by the pharmacokinetics, pharmacodynamics, as well as the non-clinical components of these studies. So this is how we differentiate between the studies that are conducted on the originator compared to the studies that are conducted on the biosimilars. Now, the studies have also shown that the TNF, binding curves of the, for example, the biosimilar adalimumab, when it was compared to the originator adalimumab, they were like superimposable, so you can see that there was no difference in the binding capacity between the biosimilar as well as the originator. On the other hand, if you look at the potency of the biosimilar compared to the originator, again you can see that the curves are overlapping on each other, which means that biosimilars are as potent as the originator, and they're as comparable as the originator in terms of binding capacity, as well as the peptide pattern of these medications is also similar, despite having different batches in terms of the structure of these medications. So let's move on to the clinical part of this talk. So we have the ADAXIS trial, which was a multi-center randomized double-blinded placebo, so controlled trial, which tested patients who were on Hyrimoz, which is one of the biosimilars of adalimumab, and against the reference adalimumab. So this study was conducted for a period of a few weeks, and then you can see here that patients were switched in a very beautiful fashion. I think this is the first time I see a kind of study where you switch patients into 4 different groups. So there is a patient group that continued on Hyrimoz throughout the study period, and then there is 2 groups of patients where they had multiple switches, starting from the reference adalimumab, then switching them to Hyrimoz, then to reference adalimumab, then Hyrimoz. And there was a 3rd group where they started on the reference adalimumab, then they were switched to the Hyrimoz, then the reference adalimumab, then Hyrimoz, then they were again switched back to the reference adalimumab. And the last, group of the study population who were continued on the reference adalimumab throughout the study period, which was like 51 weeks of the study. Then the primary endpoint was to assess like the efficacy of the medication in terms of like the patients who were having changes in their PASI 75 score. And as you can see that there was no statistically significant difference between both groups. And then when there was like the secondary endpoint was to assess like the mean change or the mean percentage change in PASI between both groups. And again, you can see here that there was no difference in the secondary endpoint between both treatment like groups. Adverse reactions, with the exception of injection site reaction, I think there were no statistically significant differences. And I think this was because of the manufacturing modification that happened in the originator over the past few years, in which they were reducing the concentration of citrate in the originator, and that's why this is the only drawback. However, I think pro management of these injection site reactions in terms of like warming the medication as well as spacing it in a way or another might help reduce the injection site reactions that can develop secondary to the biosimilar. Again, immunogenicity was not like different between these two medication groups, and the anti-drug antibodies that developed in both groups were like similar in terms of their percentages. So the conclusion that was obtained from this study is that there was no clinically meaningful difference in the long-term efficacy among patients who received continued biosimilar as well as reference adalimumab, or who switched between these two like groups multiple times. There was no relevant differences in the exposure to study treatment between treatment groups. The third conclusion was among all patients who received more than one or equal doses of study drug, there were no clinically relevant differences between the treatment groups with regard to safety or discontinuation rate, and safety profiles as well as immunogenicity were all like similar among both study groups. So that was a study on patients with psoriasis. Now let's just switch gears to a study that was conducted on patients with rheumatoid arthritis. And again here, patients were randomized either to Hyrimoz or to reference adalimumab. And then after like the end of the first period of the study, then they were switched. So patients on the reference adalimumab were switched to Hyrimoz, and patients who were on Hyrimoz, they were continued on Hyrimoz. And as you can see here that when it comes to the disease activity that was measured by DAS28 CRP, there were no statistically significant differences in the DAS28 CRP scores between both groups. Again, the mean change in the DAS28 CRP scores over like 48 weeks of the study were not showing any statistically significant difference, which indicates that the biosimilar was working in the same efficacy and potency the way that the originator was working on. And so this is basically a sub-analysis of the study results in terms of the ACR 20, 50 as well as 70. And as you can see here that there was like consistently similar response to both biosimilar as well as the originator medication group. Again, the side effects were not any different among the study group. So the conclusion that was obtained from this study is that in patients with moderate to severe RA who had inadequate response to DMARD, switching to Sandoz adalimumab demonstrated similar efficacy and a comparable safety and immunogenicity profile from the head-to-head comparison to the reference adalimumab. Efficacy was sustained after switching from reference to biosimilar with no impact on safety and immunogenicity. This is the last study that I'm presenting. This is the Dan Bio, or a study that comes from the Denmark, the Danish rheumatoid arthritis registry, in which they switched patients into either one of the biosimilars, which is Hyrimoz. So patients in the eastern Denmark, they were switched to Hyrimoz, and patients in western Denmark, they were switched to Emraldi, which is another biosimilar to adalimumab. And after like switching, it was noted that there was no difference between both biosimilar groups and in terms of efficacy, and the withdrawal was also like similar. So, and all patients in both groups achieved like remission rates at 6 months, with approximately like 50% of patients achieving DAS28 remission or ASDAS remission. Ladies and gentlemen, Mr. Chairman, to summarize my talk, I would say that although biologics had a positive impact on patients with systemic autoimmune rheumatic diseases, however, their cost remains a significant barrier to their access. And we saw that patients in Europe, about 50% of the patients were not able to have access to biological medications. Variability is an inherent feature of all biological medications, which makes biosimilars another version of the biomolecule, so i.e. not identical, but in a way or another like similar. Efficacy is sustained after switching from reference adalimumab to biosimilar adalimumab, with no impact on safety and immunogenicity. With that, I would like to thank you for your kind attention and listening. Thank you very much.